Folks. Just a little bit of housekeeping before I hand it over to Diana for the introduction here. Just to keep things running smoothly, and sometimes we do have people on pretty limited internet connections, so we'd ask that folks keep their cameras off and make sure to mute your mics. There will be time for questions at the end, so feel free to just throw those in the chat box as they come up, and we will read them out when we get to the end of the presentation here. So with that, I will hand it over to you, Diana.
Right on. Thank you, Russ. Welcome, everyone. Thank you for making the time to join us. This is our third webinar of the 2025 Winter Webinar Series. Title of this webinar is The Role of Plant Breeding in Native Seedstock. I'm Diana Boxma. I'm a steering committee member with the Grassland Restoration Forum. The Grassland Restorations Forum mission is to provide a forum for information sharing, tool development, research, and education to promote and support conservation and effective restoration of native grassland ecosystems in Alberta. As part of Grassland Restorations Forum mission to promote education, we have a few courses coming up that I want to tell you about. First one is tomorrow. It is our How to Use the Range Plant Community Guides course, and that is in Cochrane. We have literally 2 spaces available for that. So if you really want to pounce on that one, send Donna an email right now. But do remember, we also offered in September in Brooks. So if you aren't able to make tomorrow, consider it for Brooks. In Brooks is our grassland assessment training course. It's a 2-day. Day 1 is in classroom, which is what is occurring in Cochrane tomorrow. And that is the How to Use the Range Plant Community Guides. And then day 2 is a field course in Brooks there in early September. So just a lot of people kind of skip day 1 of that 2-day course. And I think you're really missing out if you do that. Like day 1 in the classroom is really using those plant community guides and how to use them really puts together the pieces of how a plant community works, and it really makes you better at your job. So you understand all aspects of reclamation, and you're just able to put all the parts together, like the DSAs, the ID, the succession planning, which plants work in which areas, and like seed mix design. It fits. So I would really encourage you to take both days of the course, or try to make it to Cochrane tomorrow if that works out for you. Keep in mind too, that I find taking that course more than once is helpful. So if you've maybe been to the field component or both days before, you consider registering for it again. So look for that early September in Brooks. You can look at our website to check that out. We'll also do our industry tour in October. We haven't announced details on that, but again, stay tuned to the website, and our fall information session will be in November in Claresholm. And then our last winter webinar of this series will be April 24th with Dr. Carol Frost. And that one is on choosing flower species for restoration of beneficial insect diversity. You can register for that, same way that you did this one. I also wanted to make note of an organization with GRF that relates to this talk, and that is the Southern Alberta Native Seed Collaborative, or SANSIC is the acronym. And this group has the common goal of improving access to local native grassland species. Sean has been a member with SANSIC since the beginning, and his background has been really valuable for the group. So do have a look at the GRF website. It's the last tab that talks about SANSIC, or be sure to either sign up for our emails and our newsletters, or just stay tuned to the website to get more information on SANSIC and our webinars and any other training courses that we have coming up. So enough of the self-promotion. I'll introduce Sean for you, and we'll get on to this. So Dr. Sean Asselin is a perennial forage grower. I'm going to start again. Dr. Sean Asselin is a perennial forage breeder with Agriculture and Agri-Food Canada. He's based in Swift Current, Saskatchewan. His program is focused on breeding adaptive native forage and dual-use grain and forage crops using both classical and molecular biology-based breeding approaches to enhance agroecosystem resiliency. Some of Sean's current projects include the development of native plant germplasm cultivars adapted to Western Canada for forage and reclamation use, characterization of the adaptive capacity of native and introduced forage legumes and grasses to abiotic stress, and landscape genomics of grassland species. Sean is going to speak to us today about how his work is contributing to more resilient landscapes by improving native seed sources. I'll let you take it away, Sean. Thank you.
Okay, thanks, Diana. And I'll start sharing my screen here and just let me know if I'm in the proper presentation mode here. Let's see. So I don't see you guys, but do you see the full screen or is it does it show the little black box around?
It's just the PowerPoint again. So it's
Like
not
the like
presentation mode.
not presentation mode. Okay, great. So I'll start then. So thanks everyone for
No,
joining
it's not
today.
presentation
Oh,
mode. Sorry.
oh, okay, sorry. Let's see. I'll switch display settings. How about now?
Still the same.
Okay, let's see. How about now?
It's still the same.
Ooh,
Oh, wait, it's changing. There you go. We got
there we
it.
go. Okay. So thanks everyone for joining today. I'll get started on my talk here. So as Diana mentioned, my name is Sean Asselin. I work with Agriculture and Agri-Food Canada at Swift Current Research and Development Center. Let's see here. There we go. There's AFC. So just a little bit of introduction on what I'm going to talk about today. I'm going to start with a little bit of context of native forages within the North American prairie. Then I'm going to move into some of the background on forage breeding that's being done here in Swift Current with a focus on native species. And then I'm going to go into a few of our current research projects on breeding native forages, as well as some other projects on improving vigor of native legumes, some work that we've just started doing on lupines, as well as some genomics work we're doing with Genome Canada on a project called GG4GHG. So starting a bit of a context about native seed availability in Canada. Historically, 24 million hectares of native grasslands were found here. And we had a decline between 60 to 99% across Western Canada since the break-in in the prairie in the late 1800s through 1930s. As a result, and I just got a breakdown between Manitoba, Saskatchewan, and Alberta there. We tend to see going across the prairies that as we go further west, we see greater amounts of intact prairie, and that's largely due to the fact that these lands are often used in grazing systems. So we have maintenance of rangelands with native plants allows them to persist. With the decline of the native prairie across North America, we've lost a major carbon sink. So in terms of mitigating CO2, the preservation and restoration of native rangelands plays a major role, and it's one of the largest things we can do in Canada to mitigate CO2, because grasslands contain about 34% of terrestrial global carbon stock. So that's the amount of biomass that's sequestered below ground, as well as the above ground biomass contains a lot of carbon out of the atmosphere. So in terms of the niche of native forages, they're valued for their late season productivity. The graph I have on the right side of the screen here is common forage species grown in Saskatchewan, as well as a graph showing native grasses. As you can see, this little diagram here shows kind of the peak in productivity in grasses and introduced legumes. And for the most part, many of our introduced species from Europe tend to have high productivity early in the growing season. But towards the middle to the end of the growing season, we enter into a summer slump of low productivity, which I've outlined here in the box. And that's really where the value of native forages is utilized, at least in the Saskatchewan or Western Canadian context, is that they tend to emerge a little bit later than our introduced species, but they tend to maintain greater forage quality, greater biomass production later into the growing season when we start to enter kind of midsummer droughts because they've adapted to the growing region. And as I mentioned previously, the bulk of our remnant prairie is preserved within rangelands. So maintaining native species within cattle production systems has benefits for biodiversity. Also, these species have evolved under regimes of fire and grazing. So it's actually beneficial to these plant populations to have those animals on the field. Animals help to break up the canopy of certain species, so we don't just have one large species overtaking a plot or a rangeland. It helps renew the system through that disturbance. Moving into a little bit of history. So we've actually been developing native grasses for forage production for quite a long time in Canada, over 100 years now. Earliest varieties were actually developed in Western Canada from primarily Saskatchewan, looking at the major species being slender wheatgrass. This is a species that early work started in the late 1800s in Manitoba, and by the 1920s, there was growing research being conducted at various Agriculture and Agri-Food Canada research stations, including the Dominion Centre in Manitoba, as well as the Experimental Farm in Ottawa. Collections were made by that central experimental farm from different sites across Western Canada and redistributed to various public sector entities, including AFC, as well as universities. And in the 1920s, we actually had 3 varieties that were developed of slender wheatgrass. AFC developed a variety called Grazer. The University of Alberta developed a variety called Freya, and the University of Saskatchewan developed a variety called Mecca. So these are very, very old varieties. You can't find them anymore, but there's just a little bit of context that we've been doing this work for a long time. And it's something that's actually, with the breaking of the prairie and immigration into Western Canada, the value of native grasses was actually a selling feature. So that top right graph or top right figure there is actually from an old advertisement in England, touting the value of the nutritious wild grasses of Western Canada. So the use of native grasses in forage production systems is nothing new here. It's actually kind of the classical or traditional state of their use or of the landscape. Moving into Swift Current. So this is where I work, Southwest Saskatchewan in Swift Current. We work on a variety of species at various stages of development, and these include warm season grasses, cool season grasses, forbs, and legumes. About 80% of my program is focused on native species, but we also do some conventional forage work, primarily in conjunction with the University of Saskatchewan, University of Manitoba, and other forage breeders across Canada. And so we do have a little bit of work on alfalfa and intermittent wheatgrass, but the bulk of my program is focused on native species. Now, what we actually do is we look at various elements of native species in terms of adaptation. This is the first step in any breeding program is to determine if the material you're working with is actually adapted to the growing season. And this is a really important characteristic in native forages. Not all populations of a given species are adapted to all growing regions. Just because a species is native to Canada doesn't necessarily mean that the genetics within that population are actually adapted to the growing region, because we do see variation within species across their distributional ranges. So a big role that plant breeding takes in the development of native seed stock is actually determining if different genotypes, different populations of native plants are actually adapted to the growing region. So we do quite a bit of adaptation work here in Swift Current. And this is really with the goal to reduce negative outcomes. When a producer purchases seed of a native species, we want to make sure it's actually adapted and it actually grows as a perennial within its intended ecoregion. And this is to reduce potential maladaptation or also known as genetic offsets, where we have failure to establish or we have failure of materials to persist. And this image in the background here, this is a population of marten nodding brome that we put in 2020. Originally, about over 200 plants were in this plot. We ended up with about a dozen plants by the end, or actually by after the first year of growth. So we had fairly severe winter kill, and part of this was due to low snow cover and Swift Current in that year. But it's just a reminder, just because a species is native to Western Canada doesn't necessarily mean that it has the genetics that are adaptive to the growing conditions. Okay, and now going on to some of the traits after adaptation, there are some agronomic traits that we do select for in native species. And this is primarily to meet some of the agronomic challenges with the harvesting and distribution of native plants. Big things that we look at are establishment, seed production, and in the context of forages, yield and quality. And as well as the managing of wild traits. Seed quality is a big issue in native plant species. Not all seed produced off a plant is necessarily viable, and there's multiple reasons for this. It could have to do with maturity of the seed, or it can have to do with high dormancy, which reduces the seed is viable, but it reduces the ability to establish in a timely manner. So it might take 3 or 4 years for seedlings to show up, or even longer. So these are some traits that we do look at, and we do select for improvements in these characteristics to make the management of, or the establishment and management of native species a little bit easier in the context of forage production. And some of the other characteristics we do look at that affect our ability to harvest seed would be shattering. So these would be with the seeds of the plant aren't maintained on the stem, so they're lost. As a result, you can get a lot of either volunteer plants, or you can get plants that a lost seed harvest. So this reduces yield, and this has impacts on the price of the seed in the end, because it's more difficult to harvest. It's more difficult to get to market. Just as an example here, this is a seed mixture from a company that we're working with for a restoration project right now. And just a little bit of comparison between the biology of the species we're looking at and the impact this has on the price point. Something like June grass, which is a fairly common species, but it's not as common as something as slender wheatgrass. It's a fairly slow to establish species. And not all ecotypes of June grass actually produce seed on a reliable basis. So it may be every couple of years, or the seed quality can vary between regions where it's grown and between populations. It's also a cross-pollinated species, so it needs a variety of other June grass plants around it so that it doesn't succumb to inbreeding depression. And it requires that those other individuals to properly set seed. So these are some of the biological factors that actually increase the price of June grass, because they affect the ability to produce seed on a consistent basis. So in this example, June grass is $67 a pound. This is contrasted with something like slender wheatgrass. Slender wheatgrass is largely considered to be the most domesticated or easy to manage of our native grasses. It's about $4.50 per pound. Some of the reasons why slender wheatgrass is easier to produce is, well, it's fairly widely adapted to Western Canada. It has easy to establish and relatively good vigor in the early growth period, so it helps compete against weeds. Less inputs are needed to produce it. The mechanical harvest is fairly straightforward. You can combine it using traditional equipment. With a lot of our native species, the price tends to go up if you start having to use specialized equipment or require specialized training to produce the seed. So these are all things that play into it. Slender wheatgrass is also self-pollinated. So an individual plant doesn't need a neighboring plant to receive pollen and to set seed. Now, the breeding for agronomic traits, as well as research and development in native species, can help reduce these costs of seed production, which is a major market barrier to the use of native species, not only in forage production, but also in the restoration landscape. And just to give you a frame of reference, alfalfa, which would be a major competitor for acreage for native species, its price point is usually about $2.30 to about $3 a pound. So it has quite a bit of a competitive advantage compared to our native species. So this is some of the reasons that we're focusing on the development of native species and improving their agronomics so that we can reduce that seed price and support biodiversity through the use of native plant seed. Now, moving into selection, I'm going to talk a little bit about some of the processes that we use to develop populations. And there's quite a few, and they tend to be species specific and depend on the type of traits that we're looking at within a population. So I'll start with strain selection and composite cultivars. So under this type of a selection scheme, what we're actually doing is just comparing populations of different native species. These can be ecotypes, so collected from different growing regions across Western Canada, different ecoregions. And we take a look at their performance within a given growing region. These can be, again, ecotypes from different growing conditions, or they can be populations that we have geo-referenced so we know where they come from. These can either be developed as source-identified seed populations outright, or multiple populations can be combined together to produce a composite population. And this was previously done, quite a bit of this work was done in the 90s to develop ecological varieties. And AFC, University of Manitoba, Ducks Unlimited, and other partners work together to develop a variety of ecological varieties for Western Canada. This type of selection is really just looking at the population level. There's really no, I would say, like true selection. It's more identification of populations and where they fit. We're not actually making any active selections to change the composition of the populations. And this type of selection takes into account the population performance, but doesn't take into account differences between individuals within a population. And the best suited populations are selected to be either developed as source identified populations, or they're mixed and increased to produce a variety like an ecovar, or ecological variety, I should say. Now, another method that we use is recurrent selection. So this is where we actually make directed selection for traits of interest. And this is commonly what we do in most forage cultivar development. Forages are a bit they're distinct compared to a lot of our other crops in that they tend to be comprised of populations of unique individuals. Something like wheat can be is highly inbred across the whole genome. So like a field of wheat, you're actually looking at genetically identical individuals across the whole field. This is different in an outcrossing species like most of our native forages or a lot of our conventional forages, is that we have greater genetic diversity. And some of the benefits of having that greater genetic diversity is greater adaptation over time because we have different buffering effects of different genotypes. And disease resistance is kind of the classical example. We can have multiple variants of disease resistance genes within a population, or variation in other traits that reduce disease incidence. And that helps with the sustainability of the population. In our outcrossing species, like forages, we tend to have a slower selection cycle. Instead of a rapid change in the population, we kind of push the population in a direction slowly over time, and until we get the end result. But care is needed whenever we're making selections to make sure that we aren't selecting too strongly, because this can have a negative impact on genetic diversity. Just an example, sometimes there's unintended consequences of making selections. And a classical example would be flowering time. Flowering time is a major life history event within plants, and it has downstream effects on things like timing of plant dormancy, timing of plant emergence. So changing or selecting for plants that flower early or flower later can have unintended consequences, because that flowering time is a major physiological signal for other linked adaptations within the plant. So like winter dormancy, that type of thing. So these are things that we look at very carefully. Going on to one of our selection cycles is mass selection. When we conduct mass selection, we're actually looking at variation within a population or a germplasm pool, and that's highlighted in blue there. Essentially, it's like taking a mixed bag of Lego, or a mixed bag of marbles or something like that, and you're selecting based on one characteristic across the entire population. So let's say you have a bag of marbles, you're selecting all the blue marbles and all the green marbles out of the population, but you don't want the red marbles. That's basically mass selection. We don't take into account the relationship between individuals when we're mass selecting. We're just looking at a given phenotypic trait or given characteristic of that individual plant, and we're bulking them all together, and that creates a new population. And from there, we can repeat this cycle of selection until we get the proportion of that trait that we want within the population. And I will note that mass selection can either be passive or it can be active. Some forms of mass selection is just taking a diverse population of a plant, growing it in a given environment, and then just taking all the plants, or the seed off of all the plants that will persist over time. So if there's differential survival, just harvesting that seed is a form of passive selection. And this is something that we observed in ecological varieties over time, because ecological varieties tend to have a broad genetic base. They tend to have adaptations to different growing regions. So we can see passive selection for that given environmental niche over time. We can also do active selection for mass selection. We typically don't practice that too much because it can result in issues relating to inbreeding. But some programs do mass selection for seed production, biomass quality, and establishment. And just to point out, mass selection can be unintentional and can be a response to production practices. This is just a paper that just came out in Molecular Ecology describing a situation with a native plant species that where they've seen changes in the morphology of the species and its genetic characteristics based on the fact that it was grown in a given area and it was repeatedly being harvested over time. And the population was starting to shift to be optimized for that production system. And ways that we can get around this is by using what's called certified seed or using seed that has labeling on its history so we know how many generations that it's been increased. Because when you have a genetically diverse population, if there's any selective advantage, we can see shifts within the population over time. And this can happen even with wild harvesting. If you're wild harvesting at the very earliest point of the flowering time of a population, you can have inadvertent selection for differences in flowering. So it's just something to keep in mind that sometimes selection is not intentional. Okay, moving on to family selection. This is what we primarily use within our program. And in the unit selection in this type of selection scheme is we look at either half or full sibling families from a diverse germplasm pool. And we often combine that with selections within families. So in this type of breeding scheme, this considers genetic diversity between families, but also variation for given traits within families. And this is a way of advancing materials under selection without being too stringent on constraining genetic diversity, because we may remove one or two families in general from the whole population, but then we're making selections within that family as well. And all those individuals are pooled together at the end to generate the next generation. And this is kind of a gentler way of making selections without being too stringent, without removing too much genetic diversity. And it's also a fairly effective way of making advancements under selection without losing all the genetic diversity in the population. Okay, so now I'll move on into some of the species we're working with here in Swift Current. Start with cool season grasses. These are species that tend to have very good early season growth and good cold tolerance. The main targets that we're looking at in a lot of our cool season grasses are for the forage market, so forage productivity, seed yield, and persistence, as well as quality. A lot of our cultivars for native plants are cool season grasses, primarily wheat grasses. There's been a long history of breeding them. And the major species that we're currently working with here in Swift Current include northern wheatgrass and plains rough fescue. But we also have populations of blue bunch wheatgrass and green needlegrass that we've done work on, and a variety of other wheatgrasses. But right now we're doing multi-site testing on northern wheatgrass and plains rough fescue to get some varieties developed. And we're looking at doing some source-identified seed work on blue bunch wheatgrass, as well as reconstructing some older ecological varieties like AC mallard of green needlegrass. Okay, moving on to warm season grasses. I should point out that last slide, that's northern wheatgrass. That's a population that we're growing in Swift Current. Moving on to warm season grasses. So this is prairie sandreed in the background here, which is a highly productive warm season grass. Our warm season species tend to have greater later season productivity. So there is some interest in the cattle industry for these species for potential for either late summer grazing or for winter grazing, because they tend to hold on to protein content and biomass later in the growing season. Unfortunately, there are some palatability issues with some of our warm season grasses. Some are stemmy or have a high silica content, which can reduce grazing or grazing use. But at the same time, in the winter months, when it's the only thing on the field or that's available to cattle, they are happy to eat it. It just may not be a preferred forage during the kind of the prime growing season. And sourcing adapted populations of warm season grasses is tricky in Canada, primarily due to the fact that a lot of our warm season grasses were expatriated due to grazing practices that occurred in the early 1900s. Warm season grasses are quite sensitive to overgrazing early in the growing season, and that was a common production practice imported from Europe in the early 1900s. So as a result, there was quite a bit of overgrazing that occurred in Western Canada, and we did see a major decline in our warm season grasses. That being said, we still do have remnant populations, and that's what we work with. The major species we're working with in terms of warm season grasses include side oats grama, prairie sandreed, as well as some smaller projects. We're doing some variety trials on switchgrass from a program out of Quebec. We're also doing some work on little blue stem as part of a few different projects. Okay, moving to legumes. So I've got the lovely purple prairie clover flower in the background there. These are nitrogen-fixing species. We're really focusing on seed quality and establishment of legumes. A lot of our legumes are small seeded and have poor vigor, so we're focusing on making them a quicker establishment or more consistent establishment. Some of the challenges we have in legume species is subspecies contain anti-nutritional compounds. So something like lupines, they contain alkaloids, which can have a negative impact on animal health. That being said, we do see genetic variation in these traits. So when we are working with most of our legume species, we're trying to focus on species that don't contain anti-nutritionals. So this includes species like white prairie clover and purple prairie clover. We're also doing some characterization work on species such as lupines for their forage potential and as well as the anti-nutritional content to see if they can be used as native forages. And then shrubs and forbs. This is kind of the least developed part of the program right now. We're just starting with conventional breeding. A lot of our forbs have a limited commercial availability, and this is largely due to production challenges. A lot of our forbs tend to have a woody growth habit, which can complicate harvesting by combine. We also have issues like in species such as winter fat, which is the species on the right-hand side of the screen there. We have male and female plants, so only half the plants produce seed. And if you don't know which plant is male or female when you plant them, harvesting mechanically can be quite challenging. So we often have to use hand harvesting for that species. The species on the left-hand side is Maximilian sunflower, and this is a native sunflower species that's quite vigorous and found primarily in the tallgrass prairie ecoregion. And it has some interesting characteristics, but it does suffer from seed shattering. So seed harvest can be tricky. It also has indeterminate flowering. So all the flowers are maturing at different times and they can shatter at different times, which again is a factor that complicates seed harvest. But these are some things that we are working on in these species. And currently in terms of shrubs and forbs, we're looking at winter fat, Maximilian sunflower, and we also have some projects on Lewis blue flax. Okay, so now I'm going to go into a few examples of some of my projects. So starting with improving the vigor of prairie legumes. So this is a project that's looking at purple and white prairie clovers, as well as alfalfa. This year, we're going to actually try out our system on winter fat. So what we've done here in Swift is we've purchased these low-cost Raspberry Pi Zero microcomputers, as well as 10 megapixel camera add-ons. They cost about $100 per unit, and we've set up greenhouse benches here that image plants at regular basis. Right now, we're doing about 4 images per day to look at germination characteristics of different populations of prairie clovers to see if we have variation in traits relating to vigor. And all this work is being processed through in-house pipelines we've developed using open source software, such as Plant CV. And from these images, we're able to extract size characteristics on seedlings, as well as shape, as well as get counts on emergence. So it allows us to calculate things like velocity of germination in different seed lots to see what's the superior seed lot. Or we can also combine it with treatments such as inoculants to see, well, what happens if we inoculate species X, Y, or Z? Does it improve the vigor and establishment? And this has a broad use across multiple species, both native and tame. And this is just an example of one of our workflows where we looked at scarified versus non-scarified prairie clover seeds. And we were looking at the percentage of green pixels within the images to look at the relative vigor of populations. So you can see here, once you get to the later days, you start to see pretty big differences in the percentage green. And we are continuing this work to include some machine learning models to pull out more shape characteristics from our images. And with this data, we can map or graph the growth rates of different genotypes over time. The above image there is a binary image of cotyledons, and the bottom image is days of growth. And we can track each individual plant over time that way, and we can make selections for early establishment and vigor. Okay, now just a few words on the lupine work that we're doing. We've started a project on Lupinus polyphyllus. So that's a native lupine to Western Canada, looking at stress tolerance and potential end use. The interest in this species is we have some sweet lupines that are grown as annual crops, and they actually make a really good forage. So we're looking at some of the perennial relatives and looking at their stress tolerance to see if we can develop some adapted perennial lupines. One of the things that we're looking at in this species is alkaloid content because that is an anti-nutritional compound. And I will note lupines are they make a good forage if you are dealing with a sweet variety. And that's really the key point. I have to note, and I cannot make it clear enough, that wild lupines can contain anti-nutritional compounds. So animals should not be grazing them unless they are sweet varieties. And a number of sweet varieties of annual lupins have been developed in Australia as well as Europe. And the genetic control of the anti-nutritional compounds is well characterized. And we do have sources of sweet perennial lupines for Lupinus polyphyllus. But at this point, we're looking at just the adaptation of different lines of Lupinus polyphyllus and swift current. One of the things that we're finding so far is lupines are quite sensitive to pH. So the water in our greenhouse really, we have very hard water here in swift current. The water in our greenhouse really did a number on lupines. But once we got them to the field, they actually established quite well. And we're going to see what materials actually overwinter, and we're going to start developing some populations based on that. And then getting at the genetics of the anti-nutritionals to develop varieties that are safe for human or animal consumption. Lupines actually have a really high protein seed, so they're being explored as a alternative to soybean in areas where soybean is not well adapted. And we currently have a project using the grow facility here in Swift Current. So Swift Current has a hydroponics laboratory. It was actually originally designed for doing salinity screening, and we've done quite a bit of work on intermittent wheatgrass and other species to develop varieties that have increased tolerance to salinity. We're starting to do more of that work on native species now, and we're looking at controlled watering, so drought tolerance of Lupinus polyphyllus. Actually, right now, that photo in the top left-hand corner is actually just from last week. We're just starting to induce drought in our populations and do some screening. And we can look at a variety of different characteristics in this facility. It's controlled by large water tanks in the basement, and we can adjust the level of salt in the water to select for materials that are saline tolerant or tolerances to different soil pHs or different nutrient content or just simply drought tolerance. So this facility plays a major role in our selections for stress tolerance in materials. Okay, moving on. I think I've got a few minutes here still to talk about GG4GHG. And as you can see there, my graphic, the G is not showing up here, but this is a Genome Canada project that I'm working on in conjunction with the University of Saskatchewan. My co-lead is Dr. John Bennett, who's a soil microbiologist as well as a plant ecologist. And we're working with the U of S, University of Manitoba, and University of Winnipeg. And we're looking at the ecosystem services associated with native plant materials. So what value does the inclusion of native plants have within a pasture or within a rangeland? So this can include benefits such as hosting beneficial insects, the type of microbial species that are associated with those different native species, as well as what impacts that those native species have on carbon sequestration. We're also looking at microbial communities as well as insect communities as potential indicators of future carbon sequestration or other beneficial ecosystem services, such as hosting beneficial insects that help reduce aphid populations or other problematic insects. Now, my role in this species is looking at genetic diversity of native species as a plant breeder and geneticist. That's kind of my bread and butter. And we're looking at the relationship between genetic, or at least in my part, looking at the relationship between genetic diversity and tolerance to drought to see how more genetically diverse populations may buffer drought in an easier way, as well as looking at the differentiation of populations across Western Canada for 5 different native species, and to develop, I guess, genetic clusters to see, do we have to develop a single population for Western Canada, or do we have distinct genetic groups, and should we be selecting within those groups for different ecoregions? So we're looking at 5 different species on this project, and the major focus is to include functional diversity. So we're not just looking all at cool season grasses. We're looking at purple prairie clover, which I mentioned previously, which is a native legume. Maximilian sunflower, which is the very charismatic pollen-friendly or pollinator-friendly species on the image there. We're looking at Lewis blue flax, prairie sandreed, which is native warm season, as well as green needlegrass, which is native cool season. And we're evaluating regional differences between the populations found in Western Canada to see if we have distinct clusters, and if those clusters should or can be developed into distinct populations that are more regionally adapted. And the main technique that we're using to characterize these populations is, well, we're using the hydroponics facility to do drought tolerance testing, but we're also genotyping the materials using a technique called genotype by sequencing. And this is a method of generating molecular markers based on the genetic code. And we can use these to determine population differentiation that might be hidden to the eye. So if a plant looks the same, we don't know how it's going to react under every condition. So the 2 plants may look the same, but at a genetic level, they can be quite distinct. And that's a place where genetic markers can really help us find that cryptic variation that can be important, not only for the health of the population in terms of genetic diversity, but also its potential for adaptation. And this is just some brand new data that I actually put together earlier this week. We just received some of our data back on purple prairie clovers, and we've genotyped collections of purple prairie clovers from across North America. That graphic on the left-hand side of the screen there, that's the distributional range of purple prairie clover. So we do have sites that we've collected from both the Canadian and American gene banks, as well as native prairie sites across Western Canada where we've genotyped. And we're starting to look at genetic clusters using genotype by sequencing to see if we have unique populations. And our initial analysis is showing that we do have kind of a central group of populations that are very closely related, which would suggest they're individual populations. But we also have some regions in Alberta and Saskatchewan that are showing up as outliers, which suggests genetic differentiation. And we don't quite know if yet if that's associated with ecoregion or other environmental variables. So we're just starting to scratch the surface there. Again, this is data that I just received and analyzed just earlier this week. So we'll find out soon what's going on in this system. I will say with some of the analysis we've done so far, we are finding some signatures that materials vary based on latitude, which is not terribly surprising. We see that in a lot of different herbaceous perennials. This is the reason why when we're sourcing native seed, we often recommend not to go, more than about 200, 300 kilometers from the source of origin in terms of latitude, because at that point we start to see differences in flowering time. The latitude has a big impact on flowering time in plants because that day length is a major environmental trigger for flowering. And on the left-hand side of the screen there, I plotted populations from Western Canada based on latitude. And you can see that there is a gradient along the principal component, in this case, between individuals. So we do see some potential that there may be some adaptation to latitude in our Western Canadian populations. And on the right-hand side of the screen there, I looked at those genetic markers and their distribution across the genome. And that big kind of skyscraper type or skyscraper pattern we see here, this is really indicative of genes that are under selection. And taking a look at the function of this gene based on the reference genome that we've aligned these SNPs to, it appears that a gene associated with photoperiod is under selection. So this may be driving the differences in populations that we see in Western Canada and across North America. But again, this is all very, very preliminary, and we still have to confirm these results. But it's just kind of interesting as a potential genetic marker that we can use to develop populations in the future or to predict how plants will perform in a given environment. So with that, I'll tie up with some thank yous. First off, I'd like to thank the team in Swift Current, my technicians, Dave Badman, Katie Hagman, general laborers, Reese Coward and Lorna Fast, who are now one's retired and one's moved to a different position, but we've got a new staff coming on board this summer, as well as my technician, Brian James, who did a lot of the molecular biology work. I'd also like to thank a variety of funders who have contributed to this project, including AFC A-Base. They've contributed through the A-Base Beef Cluster, the Living Labs program, as well as our Lupin project is also funded through A-Base. The Beef Cattle Research Council has been a huge help in funding our forage breeding work on a lot of the conventional native forages. And then I'd also like to thank Saskatchewan ADF, Saskatchewan Forage Seed Development Commission, Saskatchewan Alfalfa Seed Producers Development Commission, Sask Cattlemen's Association, as well as Genome Canada and Genome Prairie for the support on this work. And with that, I'll say thank you again. If you have any questions about the type of work I do, please don't hesitate to shoot me an email. My email is sean.aslane@agr.gc.ca, and I'm happy to take any questions that you have.
Thanks, Sean. That was great. Got one question ready to go here, and
Sure.
anybody else got one, just feel free to pop it in the chat window there. So the first one, are native forages developed for maximum foliage for grazing? Does this result in bigger plants that might not fit in well for restoration projects?
So it's actually very species specific. There, like a lot of the work in native forages has really been on establishment and making sure that things are that they're establishing well and they're persisting over time. In my program, we haven't done any major selections for like huge biomass. Like in some species, there have been instances where they've selected for like a polyploid variant of a species, which might be larger, or for more aggressive traits, and that's something that we're aware of and concerned about. So we're not doing very, very strong selection for biomass. We're focusing more on the establishment characteristics, making sure that we have good quality seed and that the seed that the producer purchases will come up. But we haven't really seen too many issues with aggressiveness. We do see that in other species. I will note some of our other forages, like especially our introduced forages, because they've had selection for biomass and productivity, they can swamp out other species. So that's kind of a balancing act that we really are aware of when developing these materials. Yeah, it's a tricky one. Yeah.
Thank you. So nothing else right now, but maybe just give folk a minute or two to see if there's anything else. Not quite. The question is, do you have varieties to growers? I'm not quite sure what that means.
Sure. So there have been varieties that have previously been released, primarily in wheatgrasses. So in terms of a certified seed, we do have a number of wheatgrasses that have been developed, primarily northern wheatgrass, slender wheatgrass, those types of species. There's also ecological varieties that were developed in the 90s. We're really trying to increase the availability of native plant materials that are adapted to different growing regions now. So we're looking at different models to do so. So if it's source identified seed or development of new ecological varieties. One of the challenges that we've seen historically with native plant materials is the maintenance and who actually owns the material. So there was previously work done in conjunction with Ducks Unlimited, where they held the rights to a lot of materials, but they moved out of native plant or maintaining native plant germplasm just due to constraints within the organization. And some of that material was transferred to AFC and maintained, and other materials went to other end users, and they may or may not have been maintained. So one of the challenges is making sure that we have maintenance of the material over time. And I see another question in terms anticipate plains rough fescue varieties be ready for increased commercially. So we have a population of plains rough fescue that we're going to start doing multi-site testing on this year. This species is it's drought limited in terms of seed production. So we've been selecting for plants that produce seed here in Swift Current, which is a very drought prone area. And we're hoping that in our multi-site testing, we will get good seed yields. From there, we're going to be moving towards registration of material. So it's going to probably take about 2 or 3 years to do our multi-site testing, and then probably another year or two after that. So we're looking at a 4 to 5 year window frame for plains rough fescue at this point. Yeah. And I should say each round of selection takes about 3 to 4 years because we are dealing with perennial species. We have to make sure that they are maintaining perenniality. And again, if we're making any selections for a trait like flowering time, those types of things, we want to make sure that there's not inadvertent consequences to those selections. So a lot of testing.
All right, so that's all we've got so far. So give folk 30 more seconds to put anything else in. All right, so I think I'll hand it back to, just a comment
Tours.
there.
So
do you do tours? Yeah.
we do for like we have done school groups before and that type of thing. Um, we also have open houses on a fairly regular basis. We will have one in Swift Current, I believe, on July 10th. It will be publicly advertised, and that will be a whole station tour. The Native Forage program will have plots on display as well as some of our facilities will be shown at that point. So there will be a major tour then. I also do one-on-one tours sometimes. It just depends on scheduling. And we also typically do a forage tour in July. I don't know if we're doing it this year because we've got the big open house. But you can expect to see some advertisement for a major open house for the Swift Current Research and Development Center early in July. Yeah. Always happy to chat about my work too. So.
All right, so with that, I think I'll hand it back to Diana for some outro stuff. And thanks everybody for joining us today.
Thanks, everyone. I just wanted to ask quick, how does one sign up for that tour or get more information to see when the advertising comes out?
Yeah, so like AFC's I guess AFC will put out like an announcement for the tour. Right now I believe it's July 10th. I'd have to check with my manager who's organizing it right now to see what her plan is for our KTT team. But there would be public advertisement for it. So Dana, I could always forward that on to SANC when the time comes, and so people can plan in advance. But right now, we're looking at, I believe, July 10th is the designated date. So, yeah.
Okay. Yeah, we'll maybe just plan for a road trip that day. Sounds kind of neat. Right
Yeah,
on.
it's a fun
Okay.
place to visit.
Right. Awesome. Well, thank you so much, Sean. Again, keep in mind, follow the GRF website and sign up for the next webinar and check out any of the rest of our courses that we have coming up. Okay. Thanks, everyone.
Thanks, everyone. Thanks for joining.